Hi, and welcome to Pharmacology of the Synapse. I'm Anna Lee, an assistant professor of pharmacology, and this week's topic is opioid receptors, pain and addiction. You've already seen a video of the pain pathway, and in this video I'll talk about pain and the opioid receptors. The opioid receptors fall into the receptor category of this organizing schematic. We'll cover the molecules that act on these receptors, their multiple downstream pathways, and the impact of opioid receptor drugs in people. The definition of pain is that it is an unpleasant sensory and emotional experience associated with actual or potential tissue damage or described in terms of such damage. Pain is usually categorized by type and duration. Some common types of pain include nociceptive pain, which is caused by damaging tissue. So an example would be breaking your arm. Neuropathic pain is pain that originates from damaged nerves. One well-known example of this is phantom limb pain, where people who have had limbs amputated still report pain in the limb that is gone due to damaged nerves. Another major category is inflammatory pain, which is caused by actions of the immune system. Pain is also described in terms of its duration. Acute pain is pain that occurs for less than 6 months, whereas chronic pain is pain that lasts longer than 6 months. These descriptions are arbitrary and are mainly to help categorize pain in the clinical setting. The endogenous ligands for the opioid receptors are peptides. There are multiple peptides produced from 3 genes, and the 3 main endogenous peptides are beta-endorphin, enkephalin, and dynorphin. You can see the genes here encode many other peptides such as ACTH, and many of these peptides need to be cleaved for processing to achieve its final form. Endogenous opioid peptides have a number of different effects, but for this video we will focus on their endogenous pain relief. You may think you don't have a source of endogenous pain relief, but one of the most commonly cited examples is the runner's high. But for most instances, endogenous pain relief is not enough, and pain treatment is required. Analgesics do not eliminate the source of pain, but instead alter the perception of pain. For example, the shot of morphine a patient receives in the ER after breaking their arm doesn't fix the arm, but blocks the perception of pain from the damaged tissue. The same concept applies to neuropathic pain. The largest class of painkillers we use today target the opioid receptors. The locations of the opioid receptors in the pain pathway are listed here. These receptors are very widely expressed throughout the central and the peripheral nervous system. Of note, the intestine and the ventral tegmental area are important areas because activation of opioid receptors in these regions can lead to constipation and addiction, which are some of the complications of using opioid drugs. The expression of opioid receptors within the brain occurs in regions that mediate both pain and reward, which will be important to remember when watching the videos on opioid receptors and addiction later on in the module. The opioid receptors are examples of a GPCR, or G-protein coupled receptor. The GPCRs are a very large group of receptors that are found throughout the evolutionary tree, from worms to humans. In humans, there are over 1,000 different GPCRs consisting of over 1% of the entire genome. The GPCRs are involved in many functions including sight, taste, smell, pain, and emotion. There are many GPCRs that are still unknown, with over 2,000 genes predicted to code for new GPCRs, also called orphan receptors. In pharmacology and medicine, GPCRs are very important as over 50% of all drugs on the market target a GPCR. We will use the opioid receptors as an example of GPCRs and explore the pharmacology, regulation, and function of these receptors. For the opioid receptors, there are 4 categories of subtypes: mu, kappa, delta, and opioid-like receptor 1. The endogenous ligands for these receptors are listed in the table, as well as some of the effects that these receptors have in the whole organism. You can see that mu opioid receptors are involved in both analgesia and euphoria. Kappa receptors are involved in analgesia, dysphoria, mood, and stress. Delta opioid receptors are involved in analgesia and learning and mood. And then the opioid-like receptor 1 is involved in stress, anxiety, learning, and immune function. For pain, we will focus on the mu opioid receptors.